Hello grade 10 students, welcome to our virtual class for the 4th quarter week 5. I'm Abigail Sarmiento, your science teacher. Our first lesson for today is about lipids. In the previous lesson, you have learned that carbohydrates are important in providing instant energy for cells. There is another class of biomolecules called lipids that have the job of storing energy for later use. Lipids are also found in hormones and cell membrane components. Foods rich in lipids. Lipids have different structural types such as carboxylic acids or fatty acids, triglycerides or neutral fats, steroids, and waxes, to name a few. Naturally occurring esters are lipids that contain one or more long-chain carboxylic acids called fatty acids. These are insoluble in water but soluble in non-polar solvents. When Aaron J. accidentally mixed oil and water, he observed that they do not mix. He was late in his chemistry class the following morning, but he was able to catch up the discussion of his teacher on lipids. His teacher explained that oil and water do not mix because they do not have the same polarity. Also, oils are composed primarily of long hydrocarbon chains. They are formed reaction between an alcohol and one or more long-chain carboxylic acids. The most abundant of the lipids are the fats and oils, also called triglycerides. Tables below shows the structures of common fatty acids. The presence of double bonds in the fatty acids lowers its melting point. At room temperature, lauric acid is solid, while linoleic acid is liquid. Table 1 is the structures of some common fatty acids, namely lauric, myristic, palmitic, steric, oleic, linoleic, linolenic, and arachidonic, and their structural formula, and then the melting point of degrees Celsius. Fats are solids at room temperature and contain saturated fatty acids. Aaron J. still remembered that all saturated hydrocarbons contain single bonds and they are produced only by animals. Examples of animal fats are lard and butter. Oils are liquid at room temperature and contain unsaturated fatty acids. Again, he recalled that unsaturated hydrocarbons contain one or more double bonds. Most oils, such as vegetable oil, corn oil, and olive oil, are produced by plants. Table 2 gives the fatty acid content of some glycerides. Can we consider fats good or bad? Depends. If you eat in moderation, fats are good sources of body fuel. They are considered good emergency food and are efficient energy storage system. However, an excess quantity of fats is not good for the heart. The reason why fats are not good for the heart is because they tend to clog arteries and overwork the heart. While carbohydrates are the main source of energy in your body, your system turns it to fat as a backup energy source when carbohydrates are not available. Vitamins A, D, E, and K cannot function without adequate daily fat intake since they are fat-soluble vitamins. If you don't meet your daily fat intake or follow a low-fat diet, absorption of these vitamins may be limited, resulting in impaired functioning. Steroids are another class of lipids whose molecules are composed of fused rings of atoms. The most important steroid is cholesterol. It is a sterol because of the presence of alcohol or the hydroxyl function group. It is found mainly in animal cells, although cell membranes of plants may contain small quantities of cholesterol as well as its major derivatives, cytosterol. Main Figure 1 is the structure of cholesterol. Figure 1 shows the unique structure of cholesterol, which consists of 4 linked hydrocarbons rings forming double steroid structure. There is a hydrocarbon tail linked to one end of the steroid and a hydroxyl group linked to the other end. Cholesterol is known as sterol because it contains an alcohol functional group, OH. Cholesterol is present in most animal membranes with varying amounts but is absent in prokaryotes. Cholesterol plays an important role in eukaryotes and especially abundant in cell membranes of animal cells. Small amount of cholesterol can also be found in the membranes of some organelles inside the cells, such as the mitochondrion and the endoplasmic reticulum. It is not only abundant in cell membrane but also in brain tissues of the nervous system. An important nerve cell, myelin, covers nerve axons to help conduct the electrical impulses that make movement, sensation, thinking, learning, and remembering possible. Studies have shown that cholesterol was found to be the most important factor in the formation of synapses, which greatly affect our memory and learning ability. Animals are able to use cholesterol to synthesize other steroids like cortisone, testosterone, and estrogen. These hormones are already discussed in grade 9. Although cholesterol is an essential lipid for humans, excessive levels of cholesterol in the blood can lead to deposits in the arteries of the heart. These arterial deposits are a leading cause of heart disease. Lemay Jr. 2000. Now, all foods have calories and different foods have different amounts of calories. Calories are provided by fats, carbohydrates, and proteins. Fats have the highest concentration of calories. On the average, that's 9 calories per gram of pure fat. Proteins and carbohydrates each have 4 calories per gram of pure protein or pure carbohydrate on the average. So, understanding the role of calories in your diet can help you balance your calories in with your calories out and help you achieve weight management goals. Our lesson 2 is about proteins. Proteins are made up of elements carbon, hydrogen, oxygen, nitrogen, and sulfur. Let's continue the story of Aaron J on his journey this time to the world of proteins. From the result of his activity, he was able to know that egg white, fish, meat, and cheese are foods rich in proteins. He learned from their discussion that proteins are found in all living cells. They are the second most common molecules found in the human body after water and make up about 10% to 20% of the mass of a cell. So whenever Aaron J eats protein-rich foods, his digestive system breaks the long protein chains into simpler substances called amino acids. He learned from his chemistry class that amino acids are the building blocks of proteins. Of the 20 amino acids found in human protein, only 11 can be synthesized by the body and 9 have to be supplied by the foods we eat. These 9 amino acids are also called essential amino acids. Adults only need to obtain 8 of them: valine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, and tryptophan. The 9th amino acid histidine is only essential for infants. Their body doesn't store amino acids, so it needs a regular daily supply of these essential building blocks. Non-essential is a slightly misleading label because these amino acids actually fill essential roles. But since they're synthesized by your body, they are not an essential part of your diet. Of the 11 non-essential amino acids, 8 are called conditional amino acids. When you're sick or under significant stress, your body may not be able to produce enough of these amino acids to meet your arginine, glutamine, tyrosine, cysteine, glycine, proline, serine, and ornithine. The remaining three, alanine, asparagine, and aspartate, are non-essential. Figure 1 shows the structure of amino acids. Amino acids are organic molecules that contain two functional groups, a basic NH2 amino group and acidic COOH carboxylic acid group. When two amino acids react with each other in an acid-base reaction, a peptide is formed. The basic amino group of one amino acid reacts with the acidic carboxylic group of another amino acid, forming the peptide, and a molecule of water is lost. This reaction shown above is classified as a condensation reaction because the two amino acid molecules join together and water is formed. The bond formed is called a peptide bond, and the product is a dipeptide because it is made up of two amino acid repeats. Longer chains are called polypeptides, and chains of 50 or more amino acids are called proteins. Proteins are characterized by their primary, secondary, tertiary, and quaternary structures. The kind of amino acids which make up the chain, the sequence in which the amino acids are arranged, and the length of the chain distinguishes the primary structure of proteins. The secondary structures of proteins refer to the coiling of the protein chain into a helix structure, formation of V sheets, or twisting into random structures. These structures are the results of interactions between R groups, H bonding, and formation of SS bonds between chains. Protein molecules are so long that they automatically coil, fold, or twist. The resulting shape is unique for each polypeptide in a particular medium at a particular pH. The tertiary structure describes the shape of the coiled chain when it is folded or hydrated in its natural state. The quaternary protein structure involves the clustering of several individual peptides into a final specific shape. A variety of bonding interactions including hydrogen bonding, salt bridges, and disulfide bonds hold the various chains into a particular geometry. The protein molecules in egg white hold an aggregate which dissolve in water. The long string of molecules unfolds once it is denatured by such agents as heat, salt, baking soda, rubbing alcohol, and etc. Another type of protein is the enzymes. It is known as biological catalyst. In grade 8 biology, you have learned that amazing action of catalyst, particularly during digestion process. These molecules speed up biochemical reactions without themselves being used up in the process. They are also highly specific. That is, they act only on certain molecules called substrates or reactants, while leaving the rest of the system unaffected. The role of an enzyme can be compared to a lock and a key. The lock will not open unless you use the right key. In the same manner, an enzyme works for a specific substrate like the enzyme lactase. Its role is to break down the sugar lactose into glucose and galactose. You must appreciate the role of enzymes in your body. Without them, chemical reactions in the body may be too slow to occur at normal condition and may affect the normal functioning of the different systems of the body. Lesson 3 is about nucleic acids. Aaron J. wonders why siblings resemble each other or how a mother and her daughters look alike. He will discover the answer as he explores the next lesson. Nucleic acids are molecules that code for hereditary traits by controlling the production of protein. Like proteins, nucleic acids are long chain of polymers consisting of simpler units of monomers. There are two kinds of nucleic acids. We have DNA or deoxyribonucleic acid and RNA or ribonucleic RNA is found in many places in the cell and carries out the synthesis of proteins. The monomers of nucleic acids are nucleotides. They are made up of three parts: a 5-carbon sugar or pentose, a phosphate group, and a ring-shaped base containing nitrogen. In this model, the sphere represents a phosphate group, the pentagon represents a 5-carbon sugar or pentose, and a rectangle represents a nitrogen-containing base. Figure 3 show a model of a nucleotide. The double helix consists of two linear strands of polymerized nucleotides that bound about each other. The two strands are held together by hydrogen bonds that form between pairs of nucleotides. Adenine, or A, forms hydrogen bonds with a thymine, or T, of the other strand. Cytosine, or C, forms hydrogen bonds with a guanine, J, or the other strand. Figure 4, a model of a double helix or DNA. Below is a summary of differences between the two kinds of nucleic acid. We have DNA, the deoxyribonucleic acid, RNA, the ribonucleic acid. Description for DNA. It contains the genetic instruction used in the development and functioning of all living organisms. Function: long-term storage and transmission of genetic information. Sugar and bases: deoxyribosugar, phosphate backbone, 4 bases: adenine, guanine, cytosine, and thymine. Pairing of bases: AT or adenine and thymine, GC or guanine and cytosine. For RNA description, it is responsible for the template and the synthesis of proteins, which in turn control the operation and function of the cell. Function: transfer the genetic information for the creation of proteins from the nucleus to the ribosomes. Sugar and bases: ribosugar, phosphate backbone, 4 bases: adenine and guanine, cytosine, and uracil. Pairing of bases: AU for adenine and uracil, GC for guanine and cytosine. The process by which an identical copy of the original DNA is formed is called DNA replication. An analogy of DNA replication is opening a zipper. As you open each side of the zipper, acts as a template for the synthesis of a new complementary strand. The result is two new DNA molecules which have the same base pair sequence as the original double helix. Proteins are the ones responsible for observable traits like curly hair, blue eyes, dark skin, etc. DNA and RNA molecules direct the synthesis of proteins in the cells. However, this is beyond the scope of this module. And that is it for today's lesson. Please don't forget to subscribe to our YouTube channel, GAITF, or log into your account at lcms.gaitf.com so that you can answer the quizzes and activities given by the teacher. Thank you for listening.